Thank you for joining us for the Image Pitch Talks. We hope you feast your eyes on the research presented by the following speakers. First up, Pascal Ibrahim from McGill University.
Hi everyone, I'm Pascal and I'm a PhD student at McGill University. When you look at this image, you might think that you're looking at cells, when in fact you are looking at something 1,000 times smaller than a cell. That comparison is similar to stacking two Burj Khalifas on top of each other versus a human on the street. This image was taken on an electron microscope, and what you see are extracellular vesicles smaller than 100 nanometers in diameter. To make it even more challenging, these were isolated from post-mortem brain tissue of a person who was diagnosed with depression and died by suicide. I study these vesicles and their cargo in the context of major depressive disorder. These vesicles are produced and released by cells, and they contain proteins, lipids, and microRNA. They act as little mail packages because they can reach other cells, deliver their cargo, and elicit a change in recipient's function. They are thought to be involved in intercellular communication, a process which is dysregulated in depression. We think that these vesicles might be transferring signals that are involved in the pathogenesis of depression from one cell to another. For that, we isolated vesicles from post-mortem human brain of 43 controls and 43 depressed suicide cases, profiled the microRNA and proteins within them, and compared between groups. We found significant differences in both microRNA and proteins between cases and controls, and some of the differences were sex-dependent. We will further investigate the function of these targets to identify possible mechanisms in which the vesicles might be involved. Another interesting aspect is that these vesicles can cross the blood-brain barrier and circulate in the blood, making them perfect candidates for biomarker discovery in depression and even other disorders of the brain. Our results allow a deeper understanding of molecular events in the brain, and overall, this field holds great potential for improving diagnostics, treatment, and prevention measures for depression and suicide. Thank you.
Thank you for sharing your work, Pascal. Next up, we have Vidya Jadrav from Université de Montréal.
A human brain is made up of billions of cells called neurons. These neurons, which comprise the excitatory and inhibitory cells, communicate with each other through junctions called synapses. This balance of excitation to inhibition is important in a well-balanced circuit in regulating sensory and higher cognitive functions in the brain. Now imagine a human brain just like a busy road in the city with junctions which herein I refer as the synapses. The communication of the traffic signals at the junction is important for efficient movement of vehicles across the road. The image shown here depicts one of the many junctions in the brain, the synapses, with the green and the red depicting the pre and the post synapses of excitatory neurons labeled with VGLUT1, PSD95 respectively. The arrowheads shown here show the co-localization of these excitatory punctas onto the cell body of Parv albumin basket cell, a class of inhibitory neuron labeled in blue, typically in a mouse brain. The number and activity of these synapses is important for efficient transmission of information across the brain, much like the communication of traffic signal at the junction. In a normal brain, this balance of excitation inhibition is well maintained. However, in most of the neurodevelopmental diseases, this balance can go awry. My study focuses on one such neurodevelopmental gene, SYNGAP, synaptic Ras GTPase activating protein. Patients with SYNGAP mutation have intellectual disability with comorbidity with autism and epilepsy. We are interested in understanding what happens to this balance of excitation inhibition if SYNGAP goes missing in the brains of these mice, mainly focusing on the red traffic signal, which is the inhibitory circuit in the brain. Do these mice have memory problem? How is this synchrony of excitation to inhibition important in sensory and higher cognitive functions in the brain? Can a mouse model provide us insights into the brains of SYNGAP patients? For much of this exciting data, please dive into my poster on understanding the role of SYNGAP in GABAergic circuit development and function. Thank you.
Thank you for your presentation, Vidya. Next up, we have Marianne Mengus from Université de Montréal.
Hello everybody, my name is Marianne Mengus. I'm working in Sophie Trump's lab in Montreal, I'm a PhD student, and today I would like to speak about this picture. Because for you, it's just many dots now. It's not really nice. Have you ever seen so many dots on a graph? What do you think they are? What do they represent for you? What they are? Do you have an idea? Because for me, they represent so many things. They represent, for now, all my thesis, because it's all my results. Not these points, but I have so many data like this. So it's not just a dot, it's a cells. Each dot are cells, and you can see the different markers, CD11B and CD45. So it's the microglia and the macrophages cells. I can isolate these two populations from the mouse brain for now, because I would like to understand the mechanisms of the macroglia cells during insert of the premature baby on the brain. So I need to take the macroglia and to isolate the population for understanding the mechanisms. So we can see on the left the quiescent macroglia. They don't receive a signal for activation, they're just screening all the environment with the many branching. And when they receive a signal, that pass to the activation phenotype and the body growth. So if the body grows, we can see the difference in flow cytometry, and they take the right population because they are so fat. And when she's so fat, they take the population in the right. It's the macroglia activated and the macrophages, because they both express the same marker. So for me, it's really important and really nice. I know for you it's like the dot and it's not important for you, because yeah, I know the immunostaining is really nice, but depending of your master and your project is not the same. So if you can learn more about my project, you can see my poster, and if you have any question, you can ask me. Thank you.
Thank you for sharing your work, Marianne. To conclude the session, welcome Anastasia Stepanchuk from University of Calgary.
Proteins perform a huge variety of functions in living systems. When their native 3D structure is disturbed, they are no longer able to play their role in maintaining cellular homeostasis. The process of incorrect protein folding and aggregation is implicated in a lot of degenerative and often incurable disorders. Alzheimer's disease is an example of protein misfolding disorders that predominantly affects the elderly. The gradual loss of cognitive function results in severe irreversible disability and eventually leads to death. Dr. Alzheimer, who first described the symptoms and pathology of the disease over 100 years ago, noted the presence of large deposits in the brain gray matter, which were later on recognized as plaques made of misfolded and aggregated amyloid beta peptide and tangles made of protein tau. Recent studies suggest that different ways that these proteins can misfold and aggregate negatively affect the brain function through a variety of pathways. Currently, we are unable to definitely diagnose Alzheimer's during life. There are no tests that would specifically detect disease-induced changes early enough to treat it. The gold standard for AD diagnosis remains post-mortem brain examination using techniques developed decades ago. And what you're looking at right now is the brain tissue of a human Alzheimer's disease case, and it's the same area visualized using two different methods. The top part of the image shows the staining of amyloid beta plaques with one of the well-established methods called immunohistochemistry. It simply reports the presence of amyloid beta peptide, which means that the areas with strong labeling are likely to correlate with amyloid plaques. In contrast, the bottom part is the same area that was labeled using our approach, labeled with small fluorescent molecules BSB and MCAAD3 that were designed to preferentially bind to and report the differences of the 3D structures of aggregated and misfolded proteins. And you can see from the get-go that our approach shows the heterogeneity of different protein aggregates, providing more information about the protein misfolding pathology in this case, by the color of fluorescence emitted from each of the 4 plaques. The old techniques can no longer advance our understanding of Alzheimer's pathophysiology, and we are proposing a new approach that reports structural variability of protein misfolding and therefore provides more insight into the disease pathology. Distinguishing postmortem signatures of aggregates in different cases can potentially correlate with different clinical manifestations during life. And perhaps with our approach, one day we would be able to diagnose different subtypes of Alzheimer's disease early enough to be able to treat it.
You have reached the end of Image Pitch Talks. Please follow the link to vote for your favorite presentation.